Well, good morning. Get my video started so you can see there's a real person here. Thank you for joining SDSU Crop Hour. My name is Anthony Bellot. I'm a soils field specialist with Extension. This week is Soil Health Week, and so we're excited to forge on with another topic in a long series of topics that Extension has had for their Crop Hour. Again, in the previous video, Matt was talking about the chat feature and the question feature. If during the presentation you have a question, please enter it in the chat or the question feature. We will be just saving those till the end of Dr. Lehman's presentation, and we'll have that discussion there. And then we're going to continue the discussion as well beyond our planned time frame. And so Matt Knippling, our Zoom technologist, will enable all of you as panelists, and we can hopefully continue this discussion about soil microbiology. So for right now, and then I'll put the scan code up for CCA credits at the end of the presentation. So for right now, I'd like to introduce to you Dr. Michael Lehman. He's a research microbiologist with USDA-ARS. That's the Agricultural Research Service. Their lab is right north of campus in Brookings there on the outskirts of Brookings. And so that lab has been there a very, very long time, and they do really innovative research. And they're focusing on soil health and crop rotations. And I see Dr. Lehman even has a proposal in for adaptive multi-paddock grazing research. So maybe getting into some livestock too as well, Mike. So with that, I'll turn it over to Mike, and we're going to learn from him about soil microbiology.
Okay, well, thank you, Anthony. I am going to first share my screen so I take all this computer stuff and get it done right off the bat. Let's see. Okay, does that look good?
We're not seeing anything yet.
You're not seeing anything yet. Okay, let me share screen. Screen 2.
There we go.
How's that working?
Looks good.
Okay, great. Well, Anthony, thanks for your introduction. It's always nice to be with our university colleagues. And I'm a soil microbiologist, so I'm going to talk about soil microbes. And what I want to do is share the unique characteristics of microorganisms and why they are essential to all life on Earth. Then I would like to share some research we've done on management practices that stimulate soil microorganisms, and close with emphasizing the importance of soil structure for soil microorganisms. Microbes are really old. The Earth is about 4.6 billion years. Origin of life, that'd be microbial life, at 3.8 billion years ago were the first organisms which were microscopic, and they had the planet to themselves for nearly 3 billion years when multi-celled algae appeared. And about roughly a half a billion years ago, microorganisms assisted plants from moving to the water to colonize the land. And it wasn't until about 100,000 years ago that modern humans populated the Earth. Well, there's a lot of microorganisms, and they very small, but together they all weigh quite a bit. So if you took the entire Earth and all the soil microbes that live deep in the Earth and in the oceans and dried them out and put them on a scale, they would actually weigh more than all the visible things like animals and trees and grass and cows and people, and you dried them all out and stuck them on the scale, they wouldn't weigh as much as the microbial life. That's a pretty amazing estimate that's been made I do want to point out that microbes aren't just confined to the first, say, foot of soil. In fact, we found viable soil microorganisms up to 2 miles deep in the earth. So they're literally everywhere. This is a block of soil with some vegetation and some animals. And you might notice here that the animals, like the birds and the worms and the moles, and the roots of the plants, they all touch and interact with the soil at very discrete points, where the roots touch the soil and where the burrow touches the soil and where the feet of the bird might touch the soil within these burrows, but they only touch it at a very discrete spot. In contrast, microbes are essentially 100% mixed up in the soil. They are as much a part of the soil as clay particles or organic matter material. They're really indistinguishable, and therefore they're interacting with soil at the finest scales possible. So it's a big difference between that and organisms that you can see. But we can take that same concept where we have the soils filled with microorganisms that are interacting at the smallest scales, and then add to it the fact that microorganisms cover inside and out every living being. And so the concept here is extended to the point where microbes are literally everywhere. They're covering every little thing. They're part of the soil. They're part of the water, and they can live under the most extreme conditions. If you consider 2 miles in the earth or in a hot spring in Yellowstone, no problem. And then this is the tree of life. This is the tree of life based on similarity with respect to a shared gene among all life on Earth. And the majority of life is microscopic. These two domains, bacteria and archaea, are both microscopic single-celled organisms that are as different from each other as they are from all the eukaryotes, which are multicellular organisms. And the only branches of life that have single individuals that are observable without magnification are these three here: animal, fungi, and plants. Every one of these other branches of life on Earth are only, a single individual is only visible with magnification. And then you talk about diversity based on genetics, but what about metabolic diversity? Almost inevitably, we talk about, okay, well, that's one microbe or that's this microbe. But really what we want to know is, what do they do? Because it's what they do that makes it such a big difference. And what they do breaks down in these types of questions. Where do they get their carbon? Where do they get their energy? What do they breathe? And what are the metabolic products? Well, with plants, most of us all know all this. We know that plants get their energy from the sun, and they get their carbon from CO2, and they breathe oxygen just like us. We also know intuitively that animals are heterotrophs, which means they utilize preformed organics like sugars or cellulose or muscle, whatever, preformed organic materials. And they get their carbon from it, and they get their energy from it, and they breathe oxygen. Of course, we all know that. But microbes have an entirely different range of ways of getting their energy and getting their carbon and what they breathe. You know, microbes can do everything plants and animals can do. They can get their carbon from CO2 and from sugars. They can get their energy from the sun, but they can also use molecules like hydrogen, methane. They can use ammonium. They can use ferrous iron, reduced iron, and they can use sulfur or sulfide as energy sources. On the other hand, they can breathe, instead of just breathing oxygen, they can breathe nitrate, or they can breathe oxidized iron or oxidized manganese or sulfur or sulfate, and even contaminants like tetrachloroethylene can be essentially used to breathe in microorganisms. And what's important about this to agriculture is if they are changing ammonium to nitrate or nitrate to ammonium with these transformations, this has a big effect on the mobility of that nitrogen in the soil, whether it stays or goes, and whether it is available to plants easily. And then some of these things like manganese and iron in the soil, a lot of elements like phosphorus or copper or zinc or any type of micronutrient will associate with things like manganese and iron oxides. Well, when these are breathed or reduced by microbes, they then become mobile. Not just the iron and the manganese and the sulfur becomes mobile, but everything that was attached and associated with them. And so it grossly affects the availability, bioavailability specifically of nutrients in the soil to plants. And so in the end, microbes are really the fundamental ecosystem service provider. Without them, there's no life. Their central roles in decomposition of organic matter and recycling of organic materials, essential roles in all biogeochemical element cycling, at global scales, at local scales, it doesn't matter. And then their relationships with all living things, whether it be plants, livestock, insects, or people. So let's take another look at some of their relationships with humans. In the last decade or so, there's been a ton of research done on human gut microbiome, basically the bacteria that live in our gut. And it's been determined that there's a strong linkage with our gut bacteria and the central nervous system, and that the microbes in our gut can be linked to some diseases like neurodegenerative diseases, but also the microbes in our gut can affect our daily mood. Seems incredible to me. And more recently, a number of associations have been made between our gut microbiota and different aging processes. So our gut microbes are linked to our aging, how fast we age, and what our chronological condition is in terms of our age. And now we find that the fountain of youth may not lie in a Florida spring, if fish are fed basically poop from younger fish, it can extend their lives, extend their lives, period. So when we're talking about crops, let's say you're going to grow some corn, often you think about how many growing degrees days we have, or how much rain we get, in particular when we get the rain, and have I applied enough fertilizer or pesticide. But have you really considered how my soil or how my microbes might affect the performance of our crop yields? And we know without a doubt that the microbes affect the performance of the humans, not just our moods, but also our aging and diseases that we might get, basically our performance. And so have you considered this? Well, we know that there's a lot of benefits from soil microbes. We've done a lot of research. We know they produce things that chemical signals that signal with plants and enhance their growth. They actually are the base of soil carbon production. This is kind of a unique, newer finding where we're finding that the soil microbes are really important in producing soil carbon and producing soil carbon that stays around in the soil. They're critical with soil structure and aggregation, nutrient availability, as I mentioned, and retention, specialized organisms that fix nitrogen, and then they may protect plants from pathogens and pests. A lot of good reasons to think about, hey, maybe I can get more out of my microbes. So the question becomes, how can management promote soil microbial life and activities? And in short, this is microbial hardship. There's no plants. The soil's been disturbed greatly. It's just not a good setting for microorganisms. In contrast, diversified crop rotations that help keep a growing plant in place as much of the year as possible offer a solution to boosting microbial activities and taking advantage of all those benefits that the soil microbes could give you. And so I want to follow this slide with some data from our research farm, where we've got a number of different crop rotations compared to a traditional corn soybean rotation. And they're all, every single one of them is no-till. So I'm going to spend a little time on this slide here. These are some of our treatments. We have the corn soybean, corn, oats, winter wheat, soybean, corn, pea, winter wheat, soybean, corn, soybean, spring wheat, pea, corn, soybean, spring wheat, sunflower. These are all no-till. Okay, and what are we measuring here? We're looking at erodible fraction. So the erodible fraction of the soils. And what you want here is a low number, a low percentage of a erodible fraction of aggregates in your soil. And what we've done is expressed all the data from these different rotations with respect to the numbers we got in the corn soybean. This is the default rotation here. And you'll see two bars, one for the corn phase, one for the soybean phase. And what you should see right off the bat is all four of these more diverse rotations produce a lower percentage of erodible fraction of aggregates. It means they have better aggregation, less prone to soil erosion, better soil structure, right? So all that's good. But you'll say, what's the deal with those two over there? Well, if you look at it, if you go and measure aggregation or erodible fraction, it actually varies over the course of the year and what crop you have in place. And so the gray bars are soybean. This is measured when soybean was in place of all these crop phases. And if you'll notice, what's unique about these two where we didn't see a lot of effective rotation, in both of those, the soybean immediately follows corn, Here, where we saw that in the soybean phase, we also saw a reduction in erodible fraction with this 4-year rotation. The soybean actually preceded corn. And here, of course, it precedes and follows. So that's hard to, you can't tease anything out there. But it's nice to know in our rotations that if we break up, we don't have soybean following corn and we have a diverse rotation, then we can lower this. Actually, I think I might have just stated that backwards. Yeah, I did. The difference here is that soybean is before corn and not after corn. That's what I needed to say. So these two, where it goes up, soybean follows the corn. When we have a better response in erodible fraction, soybean precedes the corn. So apologize for that misstatement there. So this is erodible fraction. This is the actual soils. Now I have a very similar graph, the next one. This is basically microbial activity. It's measured as short-term carbon mineralization, okay? It's the same type of graph, the same 4 rotations at the bottom. The difference is what we want here is a higher number, not lower like erodible fraction. So, and again, it's expressed as a percent of the default corn soybean rotation. And you'll see this diagram is nearly a mirror image of the previous one. And so when we reduce the erodible fraction, we're increasing the percentage of stable aggregates, and we're also increasing microbial activities. And so here again, in these two rotations where that has soybean preceding corn, in the soybean phase, we have elevated activity. We have elevated activity when soybean follows corn in the soybean phase, we don't really see that effect. And so this slide and the previous slide link the rotation effect on soil aggregation and then the rotation effect on soil microbial activity due to simply diversifying your rotation. We also have a lot of experiments with cover crops, and in the left shows one where we fall seed a cover crop, after corn or soybean harvest, and in the right is another set of experiments where we're interseeding cover crops within the corn or soybean. And I just want to show you a little bit of data. It's very similar to the previous data, but I'm going to show the data concerning this. This is a no-till corn soybean rotation, and cover crops are planted after corn and after soybean, and then the corn residue is removed from some of the plots and retained on the other plots. So that's the experiment. So this is the same graph or similar graph as before. This is for the soybean phase. This is the erodible fraction. Again, high is not good, low is better. We want a lower erodible fraction. If we have a corn soybean rotation with no cover crop and we remove the residue, we have a very high value for the erodible fraction. But if we add cover, that helps mitigate the lack of residue and its effects on the aggregation of the soil. So we can see just by having a cover crop, we maintain a lower erodible fraction. And if the residue is retained, we're good to go. We just keep our residue and we have a low erodible fraction, and whether we add cover crops or not, it doesn't really change the equation. But so the residue is more important to soil aggregation than having the cover crops. But if you don't have the residue and you add the cover crops, then you might be able to keep some form of aggregation. So then to microbial activity. This is microbial activity. This is basically the same study as the previous one, only we have microbial activity over here. Residue removed, residue retained. If we remove the residue, microbial activity is low. Doesn't matter if you add the cover crop. If we retain the residue, it's higher. And if we add cover crops to it, it's even higher. So the responses to residue removal and cover cropping are very similar for both soil structure and microbial activities. And so when we think about cover cropping or crop diversity and what it brings to the soil system, one of the things it does bring is different residues. You know, in this case, think about rye or high nitrogen vetch or clover or radish. So you bring different plant residues to the system, but a lot of the influence of diverse rotations and cover croppings goes back to the roots. And this is just a simple illustration of these four plants and the differences in root architecture. Root architecture and a diversity of root architectures, and of course a lot of roots, and are all very good things. And it makes a difference to microbes because each of these set of architecture creates a totally different set of niches for the microorganisms to occupy. And the other, probably even bigger factor is the power of plant exudates. Up to 40% of the total photosynthate that's fixed by plants is exuded from that plant root into the soil. And to me, that is just amazing, because you saw on the first slide that microbes help plants colonize the land about 500 million years ago. So for 500 million years, they've been taking 40% of the carbon that they fix from energy in the sun, and instead of building more plant biomass, they're just pushing it out into the soil, and that they haven't changed that for 500 million years. Well, the reason why is because it works. And the reason why they're pushing all that carbon out into the soil is to feed the microorganisms that colonize the area in the soil around the roots. And so what are these microbes doing down in those roots? Well, some of them are fixing nitrogen. Nitrogen is a limiting nutrient for vegetation, commonly limiting nutrient for all types of plant growth on land. It's in short supply, even though 80% of the atmosphere is nitrogen, but it's dinitrogen gas, and nothing but microorganisms can fix nitrogen gas and turn it into ammonia. There's nothing else. And interestingly enough, this ability of microbes to fix nitrogen is actually distributed far and wide across a diverse group of microbes. There are certainly the ones that inhabit nodules and symbiotic root fixers, but the free-living organisms are scattered all over the tree of life. And so this is a widespread ability that microbes can fix nitrogen, and nothing else can fix nitrogen. No animals, plants, fungi, they can't fix nitrogen. And so in terms of nitrogen in crops, if you have a soil like the first picture where you just have soil and no plants and no roots, there's some free living nitrogen fixing bacteria out there, and they're scattered about widely in the soil. But if you add some plants, just add a few plants, you have higher numbers of nitrogen fixing bacteria scattered around. You might have some little higher numbers immediately around the root, even if they're not nodulating type of symbionts. And you just generally will have a higher concentration of free living nitrogens in the soil. And then the third level is to have a legume growing. And this is really where the bulk of soil nitrogen is fixed, is within the nodules. The free living microorganisms are not thought to be a huge contributor, but a contributor nonetheless, and much more prolific if they have roots. Or if you had nodules and the plant harbors those symbiotic bacteria that fix nitrogen, then you create a lot more nitrogen. Plant uses a lot of it, and it's exuded into the soil, and it is also enters the soil through mineralization of the vegetation produced by the legumes. The other thing you might do by having a plant present as much of the year as you can and having a diversity of plants is stimulate certain organisms like arbuscular mycorrhizal fungi. These organisms form obligate relationships. It means the fungus cannot complete its life cycle without a plant. And these particular fungi, actually part of them is inside the plant. You can see those are part of the fungi that are inside the plant cells, and they extend the root system of the plant and help bring not just nutrients into the plant, but water. And they also provide protection to the plant because, again, they're obligates. So they can't reproduce without their plant hosts. So they tend to have abilities to protect their hosts from pathogens and pests so that they can reproduce. But let's move on to look at how they affect big scale things like water and air erosion of soil. or wind erosion, I should say. So fungi produce hyphae, and these hyphae act as scaffolding for small aggregates that are formed with organic matter and microbes and glue. And these mycorrhizae fungi, in particularly, not just their hyphae that work as scaffolding, but they produce a type of glue. Some folks call it glomalin. Basically, it's proteinaceous material that is kind of sticky. In fact, most microbes produce a lot of slime and sugars and things on that coat their cell walls or coat their external parts of their cells, and that promotes soil aggregation. So one way that we found that can promote the number of mycorrhizae in the soils on our research farm is by cover cropping, and that's simply having a plant growing there all year around, as much of the year as possible. This is a 3-year study we undertook. We're measuring something called Arbuscular mycorrhizal fungi propagules. This is per 100 grams of soil, so they're not super numerous. They're hard to count. But what you can see, we have 3 treatments: canola, oat, vetch, and then various combinations. And these are the numbers we counted over the 3 years. And here's no cover crop. And most of the treatments, most of the years had some increase in mycorrhizae versus none. So if you have no cover crop, you're almost guaranteed not to have an effect. If you have a cover crop, even if it's not successful, and even if it's not even mycorrhizae, there is some chance you're going to have boosted mycorrhizal numbers in the soil. So how would a producer evaluate management practices that promote microbial life and beneficial activities? And that is a question a lot of people are interested in. How can I measure it? And as a soil microbiologist, I will tell you that nothing is easy to measure, and everything is subject to interpretation. But I'm going to give you kind of a shortcut here. Let's see. Okay, so your crop management practices here, whatever you choose to do, they're going to influence the soil aggregation processes. Whatever you do here, if you can promote soil aggregation processes, that's going to turn into gross soil structure improvement that you can see visually. This actually can be hard to measure. And I mentioned earlier that if you go to a particular field, it actually will vary over time. It'll vary when it's in the corn phase or vary when it's in the soybean phase. So it's kind of tricky. When do you get the samples? How do you handle them? It's a heck of a lot of work to look at aggregates. But I think if you simply look at the production of aggregates, if you have a steady production of aggregates, you're going to create a well-structured soil. And if you have a well-structured soil, you're going to do these 4 things. You're going to infiltrate water and air. You're going to increase microbial biomass, which will support soil carbon production. You're going to increase activities that include diversity. You're going to include nutrient acquisition activities. And if you have a higher level of diversity, you're more likely to have organisms that fix nitrogen or that are mycorrhizal, or all these different types of unique organisms are more likely to be there. And here's the great thing is, if you do these things, you have more water and air infiltration, you increase biomass and activity and diversity. Those things in turn are going to produce more aggregation. And so turns out that not much soil is unchanging, that's being managed in any way. Soil is generally not just static. It's either getting better and it's getting worse, or it's getting worse. And the good news is, if you want to stimulate your soil microorganisms, all you need to do is manage for positive structure that allows air and water to go in, because microbes are just like us. They need air and they need water. Not too much, not too little, but they need a steady supply of air and water. And if you get them, they will produce more biomass, produce more activity. Again, the organic matter that they produce through their bodies, and just soil organic matter, and then the slime that's on their bodies and the fungal hyphae create more aggregates, create better structure, which infiltrates more air and water. Pretty soon, you're on this cycle of constantly improving soil, the structure, and also the soil microbial biomass diversity and activities. And with that, I guess I'd like to acknowledge one person in particular, Shannon Osborne. She's our agronomist here, and see if there's any questions that I might be able to answer. So thank you. Oh.
Okay, Dr. Lehman, thank you so much for your wonderful insights into the world we can't see. We have a question for you, and I'll read it. Oh, we have two questions. I think I got this right. Have you conducted any experiments looking about how herbicides affect the microbiome growth?
Herbicides. I'm toggling through my experience in my head, so I'm not just, I heard the question. I don't think I ever have done anything with herbicides in particular. Off the cuff, what I would say is microbes they'll view herbicides as a food source first. The first thing they'll do is look at herbicides and say, I can eat that. And the second thing would be whether it's toxic or not. And I can't really address that, but I will say that the microbes will view that first and foremost as possible food.
Okay, great, thank you. To the next question. How much of a difference is there in microbe populations when using tillage to terminate the cover crop versus no-till? And then there's a second part. There is an obvious loss of carbon with the tillage, but how does that affect the beneficial microbes?
Okay. First, I think I can say is there's been a fair amount of research done on tillage and no tillage and soil microorganisms in general. So there's a fair amount out there. And if you take an individual study, there's no smoking gun. It's not like, okay, the sky's going to fall if we till. But if you take all this literature together, you find out that reduced tillage or no tillage, you have generally more biomass, more activity, more diversity. Those are generally the things that you see. But in any one study, it won't come out and slam you on the head. In terms of whether you'd terminate your cover crops with tillage versus, I guess, herbicide, I guess right off the top of my head, I'm not aware of any studies that have done that. They're probably out there. I'm not familiar with them. And I think that's, again, every choice that's made, you have to come down to weighing the outcomes. On our farm, we terminate them with glyphosate, and I'm sure there's some reasons why we shouldn't do that. And so I think it's always going to be one thing or the other, and you're never going to be able to be to have everything just the way you want it. Um, and the one related thing is occasional tillage doesn't seem to be that bad from what I have seen. Um, that the if you have a long-term no-till situation, I think you've built the soil up so it can withstand every once in a while. I don't know what that period is, but that's been my understanding from the research and also a few observations at our research farm. Um, if you're doing every year, who knows? Not sure I got to the bottom of all your questions there, but that's within the limit of my knowledge.
Thank you. No, we appreciate that. Very good. The next question is kind of a comment first, and then the question. So the comment is, the challenge is how to convince the producer who has learned from his grandfather and father that he must do tillage. And yes, that's a social kind of economic type issue that we have. And then the question is, can we measure decreased use of nitrogen fertilizer? I'm assuming by adopting these soil health principles and ramping up the microbial activity and getting those microbes to fix that nitrogen for us.
And I hear those two questions. If it's a producer asking, I would think he'd have better insight, or she would have better insight than me on the first question. And then the second question, I'm wondering if that's more in Anthony's ballpark. I know with respect to phosphorus, there's been a few studies that have been able to demonstrate the additional bioavailable phosphorus that's been transferred to plants is allowed to have lesser phosphorus input. So I'm aware of that. In terms of, fertility studies, the ones that Anthony probably conducts or is familiar with, I know for some crops and regions, they're quite variable each year in terms of, how much you should put on, what's your response. And, I don't know how we test a microbial factor in there, how we take that away. But I think that'd be a good study. You'd have to have some land that you've done all the right things for a long time, and then you'd run those response tests on it and see how that curve might differ from land that you haven't done the same thing on. And so I'm not really aware of it being done to the point where you can quantify it into a recommendation. In terms of having to change people habits, I don't think I'm the best guy to answer that, but I appreciate it as a huge obstacle. And I'd like to hear your thoughts if you have them.
I appreciate that, Mike, because that's we're all. I think that is the elephant in the room, is that's the attraction to really building soil health, is really the need to, or the need to want to reduce our dependence on fertilizer nitrogen. But anyway, the questions keep rolling in, so let's just keep doing that for the next few minutes, and then I'll get that barcode up there. And I'm aware that there's some power outages rolling through our region to this morning, so we've been able to keep the number of participants high, so hopefully nobody's experienced that. Okay, here's the question. What benefits have you found to interseeding cover crops into standing row crops, like in the picture that you showed in your presentation?
Oh, I'm sorry. Could you repeat that, Anthony?
Sure. What benefits have you found to interseeding cover crops into standing row crops, like the picture you showed in your presentation?
Oh, okay. Yes. We have, actually, we just completed a five-year study that picture is from a 5-year study, and I don't think we've really collated the results. In the early years, we do see some positive effects on microbial activities. I guess what I would say is I don't think we've got our agronomists, we've got some hydrology data, we have the soil microbial data, and I'm responsible for the latter. And I do know there have been there's instances where the soil microbes, their activity is increased, but it's often phase specific, whether it's in the corn or the soybean phase. It's not just whenever you feel like going out there. And we've also looked at time of year, and sometimes it varies whether you take your samples in June or July or October. So I can say that the final conclusion from that is yet to come, probably within the next year as we bring all the 5 years of data together.
Okay. Then the next question, kind of a neat one, kind of a fun one here. So the question says, when town people comment to me on that fresh turned earthy smell they remember in the spring, when farmers are doing tillage, my reply is that's just the smell of your organic matter going into the air. Is that somewhat accurate?
Well, I was going to wonder if Don's on the Zoom.
I haven't seen Dr. Rakowski, so
well, I don't think it's necessary. Yeah. Obviously, you can't smell the CO2 going off in the air, but what you can smell is secondary metabolites produced by organisms that have become active and that actually are respiring, because that's what activity generally involves, is respiration. So you could say that it's linked, because those same soils or the same organisms that are becoming active, that are producing these secondary metabolites, are also the ones that are producing the CO2.
Okay, great. So what I'm going to do is I'm going to ask another question, and then I'm going to put up the CCA code for those that need to scan that. My question is, Mike, there's a lot of talk, or I see a lot of examples of folks that are growing their own, what do I would want to call it, their own hooch, their own stuff, or whatever you want to say. They're doing a variety of composting processes, and then leaching that with water to collect that tea. I seen one fellow had a big tank. He was aerating a solution of teas to increase microbial concentrations in there, I would suppose. And they're applying these things as I would call a starter, not a starter fertilizer, but a starter for their crop. It's either in furrow. I think most of them I saw were in furrow. So would you have any comments about that process? There's also been a vermiculture one where they were raising worms, and then they were catching the leachate that gone through that worm compost and applying that as a starter. So do you have any comments about that movement? It seems to be kind of strong.
Yes. I guess my answer is there's not a whole lot of high quality research that's been published on that topic. That said, I'm personally intrigued about doing some research on that. And what I'm interested in is what are the mechanisms? I mean, and it goes back to ecology. A lot of times when you apply microbes to the soil, they're not locals. They don't compete well. And if people are doing this on their own and home brewing their stuff with their own soils and maybe compost from their own farm, and they think it's having a positive effect, then I think it could be. And I'd like to do some work with that and see what are the mechanisms in terms of enhancing that. And so I guess I'm a whole lot personally more comfortable with that approach than buying something commercial and putting it on that might be organisms that might not compete well in your soils, or we still, there's a lot of variable research on how geographically relevant are certain microbes. There's research on both sides. It doesn't matter, or it matters a lot, and it depends again what you're looking at. And I suspect for something like this that it matters. The origin of the microbes might matter, and I guess I can say there's not enough really good research for me to say what the mechanism might be. I mean, if someone's doing it themselves and they're seeing positive outcomes, then I guess I would support that.
Great. Very intriguing way to do something, I thought, and made some sense, but again, I don't have any concrete evidence as well. So currently I'm putting up the CCA code. I think everybody can see that. Just want to give it just a few more moments. And I also posted in the chat how to get to our how to request extension publications. There's a Question Pro link there. And I posted a YouTube link for getting at the recordings of our crop hour sessions. So those are available as well. So you can go back and watch them again, or pass them along to someone that wasn't able to make the presentation. I'd like to talk about the upcoming crop hour presentations this week. Tomorrow we have Dr. Dwayne Beck. We're going to hear from him about doing the wrong things right, or I can't ever get that right, Dwayne. Sorry, I apologize, but anyway, he's going to be sharing his insights and wisdom and knowledge, as he always does. And so please return and help us with that. And then on Thursday, we're going to have the first type of panel discussion we've had in the crop hour. 4 of us extension folks are going to have a discussion about transitioning to soil health practices. And it's going to be centered around a publication that we produced earlier in 2020. And hopefully we generate questions, comments, and it's meant to be a panel discussion. So please join for that. We'll probably share some really intriguing pictures that you may provoke some questions as well. Then on Friday, we're going to focus on cover crops. Garrett Schaefer is going to give us a short presentation about the effects of herbicides on cover crops and the carryover effects on cover crops. And then we're going to transition again into a panel discussion specifically about cover crops. So that's the update for the rest of the week. Do we have any more questions? I'll take this down. I think it's been up long enough. We've lost a few participants, hopefully not to power outages, but they're rolling around the region. Am I missing any questions? No, I don't think so. Well, we'd like to continue the discussion into a more informal type setting. If you want to stick around and talk for a few more moments. I realize we've had really a lot of good questions already, and maybe we're all questioned out. I don't know. But Matt is now making everybody a panelist, and you can see those names coming up on your screen. And you do have privileges to turn off your mic and ask a question live if you'd like to do that. We'd encourage you to do that. Or you can enter them in the chat or the Q&A. So whatever way you want to make that work is totally up to you.